Foundation Module 3. Use Modular Integrated Undergraduate Curriculum Physiology, Dr. Sheen, CEO and founder, Sheikh Zayed Medical College, Physiology Department, Medical Superfast Simplified Physiology, Video Lecture Notes, Guyton and Hall, 14 edition, Chapter number 5, Lecture number 7, page 64. Explain physiological basis of Goldman equation. The Goldman equation, also known as the Goldman-Hodgkin-katz, GHK, voltage equation. When a membrane is permeable to several different ions, the diffusion potential that develops depends on three factors. 1. The polarity of the electrical charge of each ion. 2. The permeability of the membrane to each ion. 3. The concentration of the respective ions on the inside and outside of the membrane. It gives the calculated membrane potential on the inside of the membrane when two univalent positive ions, sodium and potassium, and one univalent negative ion, chloride, are involved. Each ion has its own equilibrium potential, which is the membrane potential at which there is no net flow of that particular ion across the membrane. First, sodium, potassium, and chloride ions are the most important ions, are involved in the development of membrane potentials in nerve and muscle fibers, as well as in neuronal cells. The concentration gradient of each of these ions across the membrane helps determine the voltage of the membrane potential. The membrane potential is the voltage difference of ions' concentrations across a cell membrane. Second, the quantity of each of these ions is determining the voltage is proportional to the membrane permeability for that particular ion. Different ions, mainly potassium, sodium, and chloride, have different permeabilities through the cell membrane. The permeability depends on the number and type of ion channels open for each ion. The higher the permeability of the membrane to a particular ion, the more that ion will contribute to the membrane potential. If the membrane has zero permeability to sodium and chloride ions, the membrane potential becomes entirely dominated by the concentration gradient of potassium ions alone, and the resulting potential will be equal to the Nernst potential for potassium. Third, a positive ion concentration gradient from inside the membrane to the outside causes electronegativity inside the membrane. The reason for this process is that excess positive ions diffuse to the outside when their concentration is higher inside than outside the membrane. This diffusion carries positive charges to the outside but leaves the no diffusible negative anions on the inside, thus creating electronegativity on the inside. The opposite effect occurs when there is a gradient for a negative ion. That is, a chloride ion gradient from the outside to the inside causes negativity inside the cell because excess negatively charged chloride ions diffuse to the inside while leaving the no diffusible positive ions on the outside. The intracellular and extracellular concentrations of these ions are maintained by active transport mechanisms like sodium-potassium pump and passive diffusion. Fourth, the permeability of the sodium and potassium channels undergoes rapid changes during transmission of a nerve impulse, whereas the permeability of the chloride channels does not change greatly during this process. Therefore, rapid changes in sodium and potassium permeability are primarily responsible for signal transmission in neurons. In summary, Goldman equation accounts for the relative permeability of the membrane to different ions, not just their concentrations. It provides a comprehensive model for understanding how the permeability and concentration gradients of different ions contribute to the membrane potential, which is important for the physiological functions of excitable cells like neurons and muscle cells. The logarithmic term in the equation reflects the ratio of the concentrations of each ion inside and outside the cell.